Good afternoon. Thanks to the organizers for the invite. I'm Michael Brubaker, Chief Scientific Officer for PolyActiva, where we're on a trajectory for transformational growth and momentum. And the momentum, as you'll see here in a few minutes, is driven by our clinical development. So there's 5 factors on our transformational growth. One is a new senior executive team in place that has years of experience. We're in the clinic and have been in the clinic, so I'll show you some 2A data on our lead asset 5108. And also, we're currently enrolling in the United States with that same asset. We've just raised $25 million. We have operations globally now, both in Australia and the United States, and we're leveraging our Prisia technology not only in the front of the eye, but in the back of the eye. This sets up a very attractive value proposition for Prisia. First, it's very sound science that will give you very precise delivery and consistent over a period of time. Focused on our customers, not only those patients who are in need, but also our physicians. And the key point I want to make here is sustainability that's driven by composition of matter patents. So each product as it's developed will last in the market time from patent protection. I want to draw your attention to the right side of the slide there on our target product profile. Each one of those attributes all add up to being able to address the unmet medical need that is long-standing in glaucoma, in patient adherence to their drops. And we all know there's implants out there today that have some shortcomings that we can resolve. Here's how our product looks. It's an ocular implant. It's based on triazole polyethylene glycol polymer that's covalently linked to latanoprost acid, and by definition then that's a new chemical entity. Zero order, no dumping, no burst, precise biodegradation that's predictable, and it's a small micro implant that fits in a 27 gauge cannula. We just locked earlier this year a successful 2A trial. It's an open label, consecutive endorsement, multi-center, and it was ran in primary open-angle glaucoma patients in 10 sites in New Zealand and Australia. Our primary objective was at 12 weeks with greater than or equal to 20% lowering of IOP from baseline. Draw your attention to the secondary objectives at 48 weeks, the same hurdle on efficacy and safety. It was 3 cohorts, an 80 microgram, 145 microgram, and a repeat dose on the 80. So we'll go next to the efficacy data. As you can see, from a washed out baseline of 24 millimeters of mercury, it drops to 6 or more millimeters over that time period through the first implant, and then even a little bit better efficacy upon the second implantation after 21 weeks. If you look at this data expressed in percent change from baseline, remembering our primary was 20%, so we met our primary by achieving 25, maintaining that 20% at the end of the first implant, re-implant at 22 weeks, and you can see we're greater than 30 on the later time points on that second implant. Here is the key data. As you all know, these implants must be safe, and we have endothelial cell density data here. As you can see from baseline on the dark colored line, all the way through the first implant, you re-implant, no negative impact to the endothelial cells, and that's carried through 48 weeks. Very comparable to the 37 eyes on drops. Expressed a little bit differently, and this is really key, and this is what the FDA looks at, is percent change from baseline cutoffs of 10%, 20%, or even more. You can see in our repeat dose cohort, there was one patient, and I can tell you that was just slightly over 10%. On the fellow eyes, remember on drops, there were 2 and then 1 even at 20. Looking at our treatment emergent AEs, 17 patients out of the 24 had AEs, 24 total AEs. Pay attention to the implant 1, implant 2. There's no difference. There's no increase in the number of AEs nor the severity. Our Presia innovation is multidimensional. We can work in the front of the eye in glaucoma. We have a 6-month and a 12-month implant in the clinic as we speak. We're doing preclinical studies for back of the eye retinal diseases. We're in advanced negotiations with a collaborator to pursue rare diseases in the back of the eye, and we're looking for cornea high-value targets as well. Thank you.
Thank you, Michael. Join us up here.